So why do we need amplification firstly? Because direct detection with probes is not possible. So I need to amplify that product. Microscopic smears are rapid but insensitive. Culture is both sensitive and specific, but it is too slow. This is the first days of the homemade PCR. We used to have these manual extraction systems. This is the thermal cycler in which there was amplification, and then there was migration and revelation on the gel dock. So there was divided into 3 steps in 2 to 3 rooms with negative and positive pressure rooms, and it used to take nearly the whole day to do the whole thing, and it also had a problem of contamination. So when contamination occurs, you could have false positive results as well. Then came the next generation real-time PCR, in which the amplification and detection was combined into the single machine, but the extraction was still manual. This is what we have today, GeneXpert. These are different modules ranging in size from 1 test to about 80 tests based on the throughput of the modules. This has been a really revolutionary technology because they have converted extraction, amplification and detection, all the 3 from 2 to 3 rooms inside a single cartridge. So everything from extraction to amplification to detection happens inside a single cartridge, zero contamination. I'll just give you a workflow of the GeneXpert. I've taken sputum as a prototype since pulmonary cox is the most common. First we do a sputum liquefaction using an agent to liquefy sputum for about 15 minutes. Then we transfer 2 ml of that material into this test cartridge. Everything happens inside this test cartridge. The cartridge is inserted into the test platform and the sample is automatically filtered and washed. Then there is ultrasonic lysis of DNA and extraction takes place. DNA molecules mix with the dry PCR agent, and then there is semi-nested real-time amplification and detection all happening inside this integrated tube. And you get the results in something less than 2 hours, 1 hour 45 minutes and thereabout. So this is that cartridge, just about this much size, from extraction to amplification to detection, all within this cartridge. The same cartridge is used for other testing, including testing for H1N1, HCV, HIV, even testing for BCR-ABL. So everything is on the same cartridge platform. So this is both CE-IVD and US FDA approved. It is highly sensitive and specific. Results are available in less than 2 hours. It's random access. You don't need to batch up. You can run a single sample. There is minimum hands-on time, and it's a totally closed system which minimizes contamination. That is most important. So the medical value is that you get the results the same day compared to both batch PCR and culture. There is a lot of time saving, and with IPAC there is also some cost saving. Just to differentiate a GeneXpert from a conventional PCR. Conventional PCR, although came much earlier than GeneXpert, has till date not been endorsed by WHO. It is moderately complex compared to conventional PCR, which is highly complex. It is 100% specific, more sensitive. It is random access. You don't need to batch up samples. In a conventional PCR, you need to do a batch analysis. The turnaround time is faster. You need biosafety level BSL-2. BSL-2 means that you just need a biosafety cabinet. Whereas BSL-3 means that you need 3 rooms. You need a negative pressure room using HEPA filters, and then you need a positive pressure room. So there is a lot of infrastructure that is required for a BSL-3 lab. It requires much less space. It can be probably fitted in the space that I am fitting my laptop. And it does not require too much skilled manpower. And the best part is there is no contamination. Now there are 2 intended uses of GeneXpert. A, it detects the Mycobacterium tuberculosis complex. And B, it also detects rifampicin resistant associated mutations on the RPOB gene. So it's a dual advantage. It is not only detecting tuberculosis, it is also screening for MDR simultaneously. However, it has not been validated for rifampicin susceptibility on patients who are already receiving treatment. It has primarily been validated for de novo patients only. So the samples that we can use today in pulmonary samples are sputum, bronchial or lavage, tracheal aspirates. In very small children, we can also use gastric aspirates. They use good sensitivity. Extrapulmonary specimens that WHO has endorsed now are CSF, lymph node aspirates. You can do an FNA on lymph nodes and run a GeneXpert on them. You can use biopsies, intestinal, endometrial, bone biopsies, etc. However, there are some samples that have not been validated on GeneXpert, and that is more important. The first is urine, because it is the most paucibacillary sample in the whole body. Plus, there is also some element of inhibition in urine. So, urine results with GeneXpert are not that good. Stool, again, for similar reasons, it is highly contaminated sample, cannot be used. You cannot use blood of any kind, peripheral or menstrual. This is very important. Rather, you should not use blood to do any kind of PCR testing, because heme is a DNA inhibitor. So, it is going to inhibit the amplification of DNA. And pleural fluid is a suboptimal sample. I have reported positive pleural fluid samples, but the ideal sample, they say, is a pleural biopsy. Again, analytical sensitivity, the limit of detection is 131 CFU per ml, which is slightly less than that of a culture. And analytical specificity, it does not detect any MOT. They have tried with 18 MOTs were tested on this system. None of the isolated were detected by the assay, so there is 100% specificity with this assay. It again gives semi-quantitative results ranging from very low, low, medium and high. This is again important for the same reason as the smear examination. This is a study by Pai et al. From India, and they did a study using all three: smear, liquid culture and GeneXpert. So on smear examination, the sensitivity was about 60%, specificity 98%. On a liquid culture, the sensitivity was 100% on smear positive and 75% on smear negative cases, with the specificity exceeding 99%. For GeneXpert, the sensitivity was 98% on a smear positive and 60 to 70% on a smear negative, again with a specificity of 98%. So it's kind of comparable to the liquid culture. So these are the WHO policy recommendation for pulmonary COX. What they say is that it should be used ahead of conventional microscopy and culture and DST as an initial diagnostic test for patients who are suspected to be MDR or patients who are co-infected with HIV. And they may be used rather than conventional microscopy and culture in all adult patients presumed to have TB. It is a conditional recommendation, basically based on resource implications. So if cost is not a concern, then GeneXpert should be used as a first line testing even before doing a smear examination. This is again a meta-analysis done by WHO. When they use GeneXpert as an initial test for TB detection, replacing microscopy, the sensitivity was 88%. And when they used as an add-on test for TB detection, following a negative microscopy, the sensitivity was 68%. Which means that if I get 100 patients of tuberculosis which I missed on a smear examination and I run all of them on a GeneXpert, I'll be able to pick 68 out of them. So I missed till 32, but I'll be able to pick 68. And when they did it as an initial test for rifampicin resistant, compared to DST, the sensitivity was 95 and the specificity was 98%. So it's pretty robust for doing MDR testing as well. This is the WHO recommendation for extrapulmonary COX. It is basically should be used as a preference to conventional microscopy and culture in initial diagnosis and testing of CSF. The sensitivity is low, but primarily the thing is that because the urgency of rapid diagnosis in such cases, it should always be used in CSF samples if you are suspecting tuberculomeningitis. And it may be used as a replacement test for samples, other non-respiratory samples, especially lymph nodes and other tissues. Lymph node has pretty good sensitivity, nearly about 80%. So this is the sensitivity specificity for different tissues. You can see that for lymph node it is about 84.9. For CSF compared to culture it is 79, but if you use it to composite reference standard it drops to 55. Because even culture cannot pick tuberculosis in CSF with that sensitivity. Next we come to the Hain genotype assays. These are primarily used to detect MDR and XDR cases. The detection is as per WHO guidelines. They have been validated in India by ICMR and they are being promoted by FIND and WHO. They run with internal quality control and it is pretty rapid. You can get the result in about 8 hours. So before I embark further, let me just define MDR and XDR. MDR is defined as TB that is resistant to at least rifampicin and isoniazid, the two most powerful first line drugs. The genotype MDBDR plus, which is actually the first line drug susceptibility testing, this has also now come in version 2 with enhanced sensitivity, is able to simultaneously detect the most important gene mutation conferring rifampicin RPOB gene and isoniazid INHA and CATG resistance in MTB isolate within 8 hours, compared with culture methods which require several weeks to sometimes months. Now in INH they have used two loci. The INHA loci is for low level resistance and CATG is for high level resistance. So if you are resistant only to INHA, you can still use isoniazid in higher doses. But if the resistance is to CATG, then you cannot use INH at all. The next is extensive drug resistance. This is defined as MDR-TB with further resistance to fluoroquinolone and a second line drug, probably amikacin, kanamycin or capreomycin. Diagnosis is even more challenging as these strains leave patients almost with very little treatment options. Again, this has also now come in version 2 where they have shed ethambutol and now only detect fluoroquinolones and aminoglycosides and cyclic peptides. So this is the standard protocol, 4-step process: DNA preparation followed by PCR, hybridization, and then there is an evaluation of the line blot. This is the WHO recommendation for first line drug. Line probe assay performance characteristics have been adequately validated in direct testing on sputum smear positive samples and on isolates of MTB grown from smear negative and smear positive samples. Direct use of line probe assay on smear negative sample is not recommended. Although the manufacturer says you can use it, but the WHO says that restrict only to smear positive samples. So this is the Hain genotype MDR for rifampicin INH resistance high and low level. So these are 5 cases. The first case is a susceptible case. As you can see that this TUB is detection of MTB, and these are 8 wild loci of RPOB, all are detected, and none of the mutant loci of RPOB are detected. So if you detect all wild types and don't detect a mutant type, it is susceptible. And similarly for CATG, it has detected the wild type but none of the mutant types. And for INH again, none of the mutant types are detected, only wild types have been detected. So this is a susceptible case. Now you look at the second case. Here you detect mutant probe 1 and mutant probe 2 for rifampicin and mutant probe 1 for CATG, but you don't detect any mutation for INH. But it is okay. Sorry. So broadly, this is how we differentiate susceptible from resistance on MDR. I'll be a little fast. Again, the same principle for XDR using basically aminoglycosides and fluoroquinolones. So this is the loci for MDR 1 versus 2. In 1 there was basically ethambutol loci which has now gone. An additional loci for gyrase B has come for fluoroquinolones, enhancing the sensitivity by 5%. An additional loci for EIS has come for aminoglycosides, again enhancing the sensitivity by 5%. So these are the difference between 1 and 2 I have already discussed. So this is the WHO recommendations for second line drug susceptibility. WHO recommends the use of second line drugs for confirmed rifampicin resistant TB or MDR TB as the initial test to detect resistant to fluoroquinolone and second line drugs instead of using a phenotypic DST because it saves time. However, there is a caveat for fluoroquinolones. The correlation between ofloxacin and levofloxacin to DST is excellent, but the correlation on moxifloxacin is not that great. So if you want to include moxifloxacin as a part of your regime, then a phenotypic DST is warranted. Just I'll take 2 more minutes. These are some new tests which are not yet available in India, but are going to be the game changer in the next coming few months in tuberculosis. This is called Expert MT-RF Ultra. So basically, the problem with tuberculosis today is that we don't have a rule-out test, which means that even the lowest LOD of about 100-150 is missing a lot of extrapulmonary and some of the paucibacillary pulmonary coughs as well. So what we need to do is to lower the limit of detection, but again not lower it to a point where it begins to give false positivity. So what they have done again here is they have increased the sensitivity and increased the higher negative predictive value of the conventional GeneXpert. And how they have done it? In fact, they have done it to an extent where they are claiming that it is better sensitivity than liquid culture. So if that is true, then it might become the gold standard also. So instead of using a 25 microliter tube, they are now using a bigger tube, 50 microliter tube, to increase the sensitivity. Also, they have used two additional loci apart from RPO-B gene. One is the IS6110 target and the other is the IS1081 target. So these two additional targets are also enhancing the sensitivity of this assay. And they claim that the sensitivity has been increased by 30 to 40%. And the limit of detection is somewhere between 5 and 50. There is a study from US which claims that the LOD is 14, but it still needs to be validated. And there is no cross reactivity with mods like GeneXpert. This is another new innovation in QuantiFERON Gold. What they have done is that instead of using one antigen, they are now using two antigens. One is sensitized to only CD4 like before, and the other is sensitized to both CD4 and CD8. So the advantage of using a CD8 antigen is that it is more frequently detected in active disease. It is associated with recent exposure to infection. I am just finishing here. And it is detected in active disease and HIV co-infection and in younger children. Another thing that has been observed is that CD8 counts begin to decline after completion of treatment, so it might have some role in monitoring as well. So a delta value of tube 1 minus tube 2 of greater than 0.6 is suggested as a cutoff for detecting recent infection in contact screening. Just to summarize what I have said, all persons with otherwise unexplained productive cough lasting 2 weeks or more or unexplained chronic fever and weight loss should be investigated for TB. There are no validated blood-based tests for active TB. Sputum is the ideal specimen for pulmonary coughs. All patients having pulmonary coughs should have at least 2 sputa specimens submitted for microscopy and or use GeneXpert, which is WHO approved molecular test. Chest X-ray is useful but cannot provide conclusive diagnosis on its own and need to follow by sputum testing. Liquid culture is very useful in diagnosing smear negative TB and drug resistant TB. And finally, drug susceptibility testing must be done on all previously treated patients and patients who remain sputum smear positive at completion of intensive phase of TB treatment and patients who have failed, defaulted from or relapsed from one or more course of treatment. Thank you. Sorry for taking so long.